Yuhan R&D Institute is a dynamic pharmaceutical research hub that develops innovative new drugs. Our patient-focused approach to R&D positions Yuhan Corporation as an emerging leader and ideal international partner in the global pharmaceutical marketplace. In addition to creating quality pharmaceuticals, Yuhan scientists study the cutting-edge platform technologies that help create tomorrow's breakthrough medicines.
건강한 혈행을 위한 오메가3 프로메가, 건조한 눈, 기억력 개선까지 프로메가.
건강도 프로답게 관리하세요.
프로가 선택한 오메가3 프로메가.
오! 종근당 건강.
Okay, hi everybody. My name is Evan Shave. I'm from Patheon, which is now part of Thermo Fisher Scientific. I'm the site manager for Manufacturing Sciences and Technology, or MSAT. I'm based in Australia, in the Brisbane facility. I've been there for 7 years. And today I'm going to present on successful outsourcing strategies for the development and manufacturing of biologics. Specifically, I'm going to be focusing on one of the newest programs from Patheon, which is a program called Quick to Clinic. And this program has been put together to really accelerate our customers' programs from development through to first-in-human trial. Okay, so as I said, Patheon is now part of Thermo Fisher as well as Fisher Clinical Services. So under now Thermo Fisher Scientific, we can offer an end-to-end solution for your biologics development program from discovery all the way through to commercialization. And within Thermo Fisher Scientific, there are already divisions existing called biosciences and bioproduction. And these divisions were already supporting our customers in biologics development, for example, in protein expression, cell biology, sample prep, cell therapy, etc. And bioproduction supported customers in cell line and media, single-use technologies, and control systems for manufacturing. And so now with Patheon on board, we're able to also offer drug substance and drug product development and manufacturing expertise and capabilities through the clinical and also into commercial. And then at the end, Fisher Clinical Services can get involved and deliver the clinical trial packaging and labeling, and also logistics to get your drug product to the trial site. And so this really is an end-to-end offering now, from early discovery all the way through to commercialization. Looking just at the Patheon biologics global sites firstly, we have sites for drug development, for drug substance. We have sites in St. Louis and Princeton in the US. And then for manufacturing of drug substance, we have GMP sites also in St. Louis. We also have a site in the Netherlands, in Groningen, and there's also a site in Brisbane, Australia for clinical and commercial manufacturing. For the drug product, we have 3 facilities. There's one in the US, in Greenville, North Carolina, and there are 2 in Europe, in Italy, Monza and Ferentino. And so I want to highlight some of the key capabilities of our network. Firstly, at the very beginning, we're able to offer cell line development services, and we're really focusing now on high throughput techniques such as some new equipment called Beacon from Berkeley Lights, which can really speed up your cell line development. Also, the Amber 15 platform to help with the early stage cell culture development. All our manufacturing sites can offer fed batch and perfusion types of manufacturing. And also, the manufacturing sites are all set up with a flexible manufacturing in mind. And so that means we use multiple single-use platforms at all our sites. We're able to accept any cell line into our manufacturing facilities that are mammalian-based. And we're able to bring in the projects at any stage of development. Whether it's early or late stage, we can accept them into our facilities, and we can also transfer them out if you need to move your project sometime down the track. In terms of expertise, our network has done over 180 tech transfers since 2014, and since 2017 we've done over 72 process development projects. And in terms of expertise with different molecules, we've done a lot with monoclonal antibodies, about 70%, but we've also 30% are also other complex proteins. So that might include FC fusions, recombinant proteins, enzymes, for example. And finally, with our experience, we've got over 11 years of commercial manufacturing experience. And currently we have 4 commercial products running in our facilities. 2 of them are running out of the Netherlands site in Groningen, and 2 others are running out of the St. Louis site in the US. Last year, across the 3 manufacturing sites, we produced over 100 GMP batches. Okay, so now looking at approval of biologics, and the approval of biologics is really increasing year on year. This graph shows over the years what percentage of new drug approvals are biologics. So the dark blue bars are the percent that are biologics out of all new molecular entities approved. And you see that at 2017, it was hovering just below 30%. And year on year, it is an increasing trend. But also looking at these numbers, we're seeing more and more products that are being put into a breakthrough therapy category. And so 17 of the 46 novel drugs that were approved in 2017 were designed as breakthrough therapies. And then 6 of these 17 were actually biologics. And so this trend of increasing breakthrough therapies means that our customers who are involved with these sort of drugs, they're looking for faster and faster ways to get to commercialization, because the regulatory agencies are giving them pathways to get there faster. And so we saw that as an opportunity to offer development and manufacturing services to get them there. And so we're also hearing a lot from the market and from our customers and potential customers about what they need right now. And these are some quotes we've heard recently, such as, I need to complete my first in human trial as soon as possible. I need to complete data set for my IND submission. I have multiple assets that I need to quickly screen. Or I need supplemental material for my IND enabling tox studies. And so all these comments are really saying, I need material and I need it quickly. And so, we're also hearing this from all companies, not just small and emerging companies, but also mid and large sized biopharma. And so because of this and the trends we're seeing, we wanted to introduce this faster quick to clinic program. So the goal is to get an IND approved quickly and cost effectively. However, the one thing you want to avoid is getting a clinical hold on your program. And so there is a balance. You don't want to go too quickly where you have gaps in your process, and it can lead to problems and that'll end up delaying you overall. And so if you look at the average time from DNA to IND approval, if you have a clinical hold, you could be looking at up to 29 months. This is what we've looked at in the market. And then without the clinical hold, it's about 17 months. And so there is a big impact on your program if you end up having a clinical hold put on you. And the types of issues that can lead to clinical holds are listed on this side. And that could involve incomplete target product profile, or an inadequate manufacturing procedure is outlined, or you may not have properly characterized your master cell bank, or you could be missing some viral clearance study data, or there could be some sort of product quality issue which is going to lead to this clinical hold situation. There are some other key things to consider as well when you're moving into developing your program. For example, it's critical that you understand the main milestones that are going to be coming up soon. So how long is it going to take to do your CDMO selection? How long will it take to negotiate the service contracts? And then once you have those things in place, how long does it take to go from the gene through to IND filing? And it's good to be thinking early on about the trade-off between speed and process robustness, and navigating some of those obstacles I mentioned. And really, it's about having a phase-appropriate approach to your development and manufacturing. Something we commonly work with customers on is how much material they need to support the many different studies that are going to go into an IND filing. And so, for example, For tox study, we help advise customers on how much material you need. That also might include the viral clearance study. And you have to consider things like number of patients, the dosage and injection volume, and then some of the side studies, such as stability studies, reference material, retains, and also any supplemental tox studies. So it's very important to plan the right amount of material you need, because that directly impacts what sort of scale you need to be operating at the various stages of your program. So let's have a look at the Quick to Clinic program now from Patheon. From our market research, we found that a traditional phase 1 program was taking approximately 22 months to go from DNA to released drug product. So with that in mind, we had an aim to hit 16 months from DNA to released drug product. So that's vialed product that's released. And to accomplish this reduced timeline, we had to do many of the things shown here, such as firstly leverage our experience with monoclonal antibodies. We also relied heavily on platform processes, which we've developed over the years. And then in terms of the workflows for drug substance and drug product, we looked to do as much in parallel as possible. And that also includes a lot of automation. And so I've already mentioned the Amber 15 and Beacon systems. We look to use pre-qualified vials and contact parts enclosures for drug product. And throughout the development program, we always take a risk-based approach. And finally, with the documentation, we heavily rely on standardized templates to speed up the document drafting and approval. So let's look a little bit closer here at the Quick to Clinic program. You can enter the program two ways. Firstly, you can come with a gene, and then we will perform the cell line development for you before moving into bench scale development. Or you can come with a clone already selected. That might be a research cell bank, and you might already have selected your media and feed strategy. And that's fine, that can feed into the program still. And then what do we use? If we're developing the cell line, then we'll use the Thermo Fisher CHO-S platform. And then we will then use the accompanying Thermo Fisher media and feeds, and also the commercially available raw materials throughout the program. On the other hand, if you come with your own, then we'll work with your media and feed, and then incorporate any of our other already approved raw materials to help speed up some of that raw material ordering process. And then what do we do? So if cell line development's involved, we'll do that first. And then for both options, we'll then enter into development at bench scale. And this will include some Amber runs to help speed up the identification of the best media and feed mix. We'll perform formulation screening for the drug substance, analytical method establishment, and then we'll produce tox batch, GMP batch, viral clearance study, and stability testing study. And what you get at the end is early non-GLP tox material as quickly as possible. You get released drug substance and released drug product. So normally we'd expect to produce at least 1 kilogram of drug substance, and that assumes your cell line is producing around 2 grams per litre, for example. You'll also receive 1 month minimum stability data for the IND and templated quality reviewed reports. And finally, as an optional extra, we can also add on the clinical trial packaging and labelling if that's required for your program. So looking at the timeline and sequence of events, the process development will take about 10 months, and then the production for drug substance and drug product is usually around 6 months. And there's a tech transfer in between from the bench scale development into one of the GMP manufacturing sites. So starting off with the cell line, as I said, if you come in needing that, it'll be on the CHO-S platform. And then moving into upstream and downstream process development. For the program, we usually offer 3 lab scale bioreactors, and we assume the purification can be done in 5 purification steps. Once the process is worked out well at the bench scale, we go into a confirmation run, which is usually at the 12-litre scale. And at this point, we're essentially locking the process for which we're going to transfer to pilot and then manufacturing. And so from the confirmation run, we can also provide the interim reference standard. And then this sets up the manufacturing in the pilot facility at either a 50 or 200 or 250 litre scale. And the material that comes out of this batch would be used for viral clearance studies and also your animal tox studies. And then from the pilot, we can move directly into one of the GMP manufacturing sites. Typically, that would be run at a 500 or 2,000 litre scale in a single-use bioreactor. And then this material is what goes into the DP site for filling and ultimately ends up in the clinical trial. Whilst these activities are taking place on the process side, we also have analytical scientists working on method evaluation and method qualification. They might be involved with a liquid formulation study. And they're also generating the interim reference standard and master reference standard to support the stability studies. And so all along, we're using a phase-appropriate approach here in terms of integrating analytics into this program as well. So formulation development is also a key part of the program. If you are coming into the quick to clinic program, it's going to be with an IgG 1 or a 4, and we don't need to do formulation development. Instead, we do formulation screening. And so we have a number of formulations we already have experience with, which we know will very likely work for an IgG. And so rather than doing full development, we'll just screen some of these formulations that have been shown to work in the past and quickly identify one that'll work. And that way it's taken off the critical path as you're approaching process lock. If you do need to do formulation development, we can also support that. And it'll just spend a little bit more time in development, where we're going to be looking at different concentrations of salts and buffers, additives such as surfactants, and then we'll confirm the stability with a force degradation study as well. So if we take a look at the whole timeline now for the quick to clinic program, you can see on this slide, beginning at cell line development, we're saying that it's going to take around 5 months, and then analytical development and process development start occurring in parallel, also including formulation development, and that gets us to a point where the process is locked well enough to run a toxicology batch. This is run in a non-GMP facility, and then we transfer it to the GMP facility for the drug substance batch, which brings you out to about 14 months. And then finally, we fill into the drug product vial to end at 16 months. And so going through all these activities, we're working in different locations sometimes, so, and also different departments within one site. And so communication and collaboration are key to making this sort of a program work, and also risk management as you're moving through this timeline. So I mentioned before about the use of high throughput to try to accelerate some of our steps during development, and I just want to highlight one of the new pieces of equipment we're evaluating right now, which is called the Berkeley Lights beacon system, and it's used to accelerate the cell line development and clone selection part of the process. And so the Berkeley Lights system uses microfluidics to move cells in and out of microfluidic chambers. And so you can see in this picture down here, these are the microfluidic pens on the chips. There are 1,750 pens on each chip, and you can place a single cell inside the pen and then expose it to different media and different feeds, and then monitor the growth, titer, and clonality using this system. And so the two main benefits of this system is, firstly, we expect the time to greatly decrease from 5 to 6 weeks that it currently takes using standard techniques down to about 5 days to select your lead clone. And the second and maybe more important part is that we're able to screen many more clones with this system than you can with a traditional approach. And we're able to screen for titer, growth, and clonality, and this greatly increases your chances of finding that rare producing clone in your transfected pool that's that high producer. And so that's really the key benefit of this system. And right now it's under evaluation in our labs, and we expect to start incorporating it into our programs in Q3 or Q4 of this year. And as I said, that will shave off some more time that's currently on our quick-to-clinic program. So in summary, for the quick-to-clinic program, there's two ways to enter. You can either come with a gene, or you can come with a cell, a clone already identified, a cell bank. And if you come with the genes, it's about 14 months to release drug substance. If you come with that research cell bank, it's reduced to about 12 months. The program is very flexible. We have very flexible licensing terms. And if you come with your own cell, then of course there are no licensing terms that we're involved with. We're employing a lot of high throughput screening technology to achieve these fast timelines. We're providing GMP batch release documentation, drug substance, formulation development, released drug products with stability data, and also clinical trial packaging, if that's required, that can be supplied. All this can go into your IND information package. So this quick-to-clinic program is really focused on monoclonal antibodies, and we recognise that these take up about 50% of all the drugs that are out there right now. However, there are also a lot that don't fit into this standard monoclonal antibody bucket. There are coagulation factors, enzymes, and fusion proteins. And so we recognise that these proteins don't fit standard manufacturing platforms. And so we need to come up with other ways of developing them, but still with speed in mind. And so, for example, we see a lot of FC fusions coming through our development lab at the moment, also bispecifics, fragment antibodies, and also just other recombinant proteins. Often these require more than 3 chromatography steps to properly purify. They're also sometimes unstable during the purification. This might require chilled steps or continuous processing. Usually they have lower titers, and often they have unpredictable performance on some of the standard resins we use. And so because of this, that we unfortunately can't just apply a standard platform to some of these more uncommon products. However, we can apply some of the same strategies that we use in the quick-to-clinic program to speed up some of the development steps. And really, this is around the use of high-throughput screening and applying it still to other molecules. And so we've been using robotics to screen resins, and usually it's the polishing steps of your process that you have to screen and modify to tweak the platform process to make work for these more difficult molecules. And so we use design of experiments to screen for selectivity and impurity removal, and then also if there's opportunities to do high throughput testing on the analytical side, we also do that. The example down the bottom here is from a design of experiments study where we were looking at yield and a low molecular weight impurity and the clearance across a particular resin as a function of pH and sodium chloride in the elution buffer. And so from a couple of days' work, the PD scientists can very quickly see where the product and where the impurities are moving and what the impact is on yield and purity, and that very quickly identify the right resin and conditions to use on some of these more difficult molecules. So as I said, we're seeing a lot more FC fusion molecules, and so we have put a fair bit of effort into trying to develop what we'd like to call a platform for FC fusions. In 2017, we published a paper in Biopharm International on development of purification for challenging FC fusion proteins, and it uses many of these techniques I'm talking about. FC fusions often have the problem with them associated with product-related impurities such as fragments or aggregates, and this can be as high as 20% coming out of the bioreactor. In addition, they're sometimes not stable at low pH, and so to address this, we've firstly on the low pH, we often use detergent virus inactivation rather than low pH virus inactivation, and so that's certainly an option we use for these FC fusions. And then for the polishing chromatography steps, we ended up looking at the use of flow-through mode in a cation exchanger and hydrophobic interaction chromatography resin to effectively remove those residual proteins, impurities I mentioned before, and still get decent yield. And then the third chromatography step was a mixed mode bind and elute step. And so even though we had to tweak the platform process that we used for the quick to clinic, we were still able to use a lot of the same techniques to get there quickly. And this process has shown to be worked for several FC fusion molecules. So no matter what type of program you decide to use, whether it's a standard process development manufacturing over the standard timelines or a more streamlined quick to clinic approach, as the drug developer, you need to be thinking about these things from the very beginning of your program. So at the very beginning, you have to be thinking about manufacturability. And this might be looking at the actual sequence to make sure that there are no problems in the sequence that can impact stability down the track. In terms of process development and characterization, the question is how much is enough at different times of your program. Cost of goods, this has to be considered early on in your program as well, because the raw materials and consumables that you select at the bench scale for your program often end up stuck with them in your commercial process. So it's worth thinking about that early on. Manufacturing strategy and forecasting. This certainly is important in terms of what scale you're going to need to be operating at. And that often drives the decision about whether you're going to be using single-use bioreactors or stainless steel bioreactors. If you get all these things right, you're going to end up with a successful program. But this is, of course, if you're going to be outsourcing your program, you need to be thinking about your CDMO. And these are some of the key things you need to think about when you're selecting your CDMO for outsourcing. Firstly, track record. They need to have a good quality and performance track record. They need to be able to work with you and share their technical and regulatory experience to help your program. Flexibility. Your CDMO needs to be flexible in terms of the equipment they offer, the sort of technology they can use, the raw materials they're going to use for your process, and even the timelines. Are they flexible with timelines depending on what sort of program you want to run? And also important is the license fees. Do they have flexible arrangements on, for example, a cell line, where it could be very difficult to move from certain providers of cell lines if you wanted to move your program from one supplier to another manufacturer. Does the CDMO have established business processes? Project management and tech transfer are key business processes that you have to get right, and they can make the difference between a successful program or not. Finally, communication. Most CDMOs operate in a global world, and so they need to have cultural awareness, and they need to be using a partnering mindset when they're working with their customers. And so, again, getting all these things right will lead to a successful partnership and a successful program. So that's the end of the talk. Thank you very much for your attention. I'd like to just highlight our booth over at F25 and the local business development executives here in Korea, Austin and Kail. Please come by after the talk if you have any questions. And I'd also like to take any questions now if there are any. Thank you.
Thank you very much. Thank you very much for your great presentation. So any other question and comments? As he mentioned, he has on booth here. Okay, you have a question, right?
Thank you very much for the presentation. My name is Dennis, and I work for the company Yuhan in Korea.
Right.
And I have one quick question about the IP protection. Obviously, you are receiving some technical information from your sponsors, and how you are managing those IP-related documents or data.
Right. Yeah. So we treat all our customers' information with the highest confidence. So there's no sharing of that data, all that information. In terms of that IP, that always ends up being owned by the customer, and so we don't take ownership of any of that as part of taking your program in for development or manufacturing. Of course, yeah.
So yeah, it's general. In a high level, I understand where you're coming from, but for example, someone who is in the BD, business development, they receive those technical information first, and then that information will be transferred to either R&D side or manufacturing side for the further discussion.
Sure.
Then how those information is it all encoded or how you manage those information transfer?
Yeah, sure. So our general way of working when we're moving, we need to share documents from around different departments and sometimes different sites. If it's confidential, we will operate within a project SharePoint. And so within that SharePoint, that's password protected. And so only the project members who are approved by us and also by the customer can have access to that SharePoint. That's usually established at the beginning of a program. And as you said, at the beginning, there's transfer of information. But then we also keep using that SharePoint for sharing data that's coming out of our labs and manufacturing back with our customer. Thank you.
Okay, so now we'll finish this presentation. Thank you very much.
Thank you.
Thank you.
아기에게 라투핏 핏핏핏핏 아이들도 라투핏 핏핏핏핏 우리 가족 라투핏
맞춤형 생유산균.
내 몸에 핏 라투핏 종근당 건강
당신이 밀려오는 두통을 참고 있을 때 당신의 친구들은 밀려오는 음악에 몸을 맡기고 있다.
두통? 참으면 당신만 손해. 참지 말고 즐겨라. 펜잘.